I get to have the fun pleasure of introducing the 2023 Young Investigator Award winners, Dr. Anna Schapiro and Dr. Freek Venere. I've been practicing, so hopefully good enough. So I'm just going to briefly introduce them and hand over their awards, one after the other, and then we'll go into the talks after that. Well, you can save your questions for the talks till the very end. You can find them afterwards. So we're just going to do the talks and then end the session after that. So without further ado, I'm going to start by introducing Dr. Schapiro. So Anna started off fast out of the gates, winning research awards already as an undergraduate student at Stanford, where she worked with Jay McClelland. Then supported by an NSF graduate research fellowship, she completed her PhD at Princeton with not one, not two, but three advisors, three taskmasters, Nick Turk-Brown, Ken Norman, and Matt Botvinnik, before she moved on to a postdoctoral fellowship with Bob Stickold at Harvard Medical School. And she started her own laboratory in the psychology department at UPenn in 2019. So one motivation for this award, and a factor that played into the committee's decisions, is that Anna's research has been really remarkable in the way that she's really seamlessly integrated computational modeling with really clever experimental studies of behavior, neuroimaging, and studies with patient populations, and really put those things together in a very uniquely seamless way to address fundamental questions in the cognitive neuroscience of episodic memory and statistical learning. So in merging these approaches, the computational and the experimental, her research has really upended some very long-held assumptions about divisions of labor between the hippocampus and the neocortex in episodic versus statistical learning, revealing functional distinctions entirely within the hippocampus among distinct circuits that had yet to be appreciated. And to quote one of her nominators, given how intensively our field has studied the hippocampus, making a genuinely new contribution to our understanding of hippocampal involvement in learning, as Anna has done, is an astonishing accomplishment. And her laboratory continues to make groundbreaking discoveries using this integrated modeling and experimental approach, yielding many fundamental insights across broad conceptual areas in the memory field, including statistical learning, categorical learning, sleep-related memory consolidation, and a variety of other areas. In addition, Anna is described and well known as a committed and caring mentor, who is I'm quoting some of this, a tremendous asset to the fields of memory research, cognitive science, and computational neuroscience through her role as an editor, an organizer of meetings and major conferences, her contributions to teaching and public outreach, and being, quote, one of the best people in the world with whom to share, debate, and refine ideas. So given all of that, it is a true honor to present this year's award to Dr. Schapiro. Come on up. Don't forget this. It's not a giant check, but okay. And then moving on to, it's my equal pleasure to introduce Dr. Venere. Freek received his PhD in cognitive neuroscience from the Donders Institute at Nijmegen, where he was mentored by an outstanding team, including Eric Maris, Floris Delange, and Ole Jensen. He then moved on to an incredibly productive postdoctoral fellowship with Kia Nobre at Oxford. And since 2020, he's been a principal investigator and an associate professor in the Institute for Brain and Behavior in the Department of Experimental and Applied Psychology at Vrije, okay, sorry, University Amsterdam. One of the main factors in the decision-making for this award is that Freek's research has been really remarkable in the detailed ways that it integrates the constructs of attention and working memory with research on anticipation, timing, and action to understand how the brain prepares for upcoming behaviors. And one of the key strengths that's noted and is obvious in his publications is his ability to take the really conceptually and practically thorny problem of behavioral planning with many processes that are happening at the same time that are poorly defined and hard to measure, and take all of that and break it down via advanced behavioral and neuroscience methods into very clear and concrete constructs with very identifiable neural correlates to really try to get a handle on how this complex system works. And this has allowed him to, capture proactive, dynamic, and interrelated nature of cognitive processes and their neural basis in very unprecedented ways, leading to many landmark theoretical insights and methodological approaches relevant to many aspects of adaptive cognition and decision-making and action planning. He's been so productive in this very difficult area because of what has been described and is apparent in his publications as an uncanny ability to break open new experimental questions with elegant and ingenious task designs, and to show methodological innovation and leadership across a wide array of cognitive neuroscience approaches. In addition to all of this scientific prowess, Freyk is described as a giving colleague who loves exchanging ideas, collaborating with those around him, and organizing meetings for the greater good, with a value to the field that greatly surpasses his impressive and significant scientific papers by enriching the fabric of science with his generous, collaborative, and congenial spirit. So it's a true honor to present this award to Dr. Venere. Please come on up. Okay, and now what you came for. We'll launch right into it, so come on up.
Okay, hi. Thank you so much for that introduction. I've sat in this audience before in young investigator talks in past years and just been wowed by all of the amazing science that these people present. So I'm really very honored to be here and to get to share our work with you. I'm also really grateful to be here because, as some of you know, I was diagnosed with breast cancer in the fall, and when I learned that I was going to be receiving this award, I was recovering from surgery and actually didn't know yet if I was going to be able to travel and be here. And it turned out that the surgery was very successful and I was able to come. I didn't need further treatment, so beyond being the honor of receiving this, I'm also just really happy to be here. Thank you. Thank you. And part of the reason that I wanted to say that is that I really benefited from the amazing support of breast cancer survivors in our field who helped me through that journey. And if anybody is facing something similar and feels like it would be useful to talk, I really would truly be happy to talk to you. Okay, so I want to start by thanking my mentors and my collaborators over the years, so undergrad and PhD and postdoc mentors, and all these different people I've been able to work with over the years, many of whom have also served as unofficial mentors to me. I'm incredibly grateful for all of their support and for doing fun science with me, and I certainly would not be here without these people. And I also want to thank my amazing lab. These are the people that make this job fun and worthwhile, and they're, of course, the people that did most of the work that I'm going to tell you about today. So thank you, lab. Okay, so I'm going to start with this really general question. How does the brain store new information? And I'm going to define two ends of a spectrum, from localist to distributed representations. And these terms are used in very different ways, so I'm just going to start by defining what I mean by these terms, and I'm going to illustrate them in terms of how they play out in these little neural network models. So in a localist representation, or in the memory literature, we call it a pattern-separated representation. Some input comes in from the environment. There's some pattern of activity in this internal representation that will that reflect that information. Some new information comes in from the environment, and you make sure that there's no overlap. A completely non-overlapping population of neurons is going to represent that new information. as opposed to a distributed representation where you allow there to be overlap in the different inputs from the environment. So Jeff Hinton said a distributed representation is where each computing element is involved in representing many different entities, as opposed to a localist representation where each computing element is involved in representing just one entity. So distributed representations have been crucial to the success of neural network models, both in machine learning and in their long history of success in explaining cognitive phenomena. They're extremely good at learning structure and data when they're paired with a good learning rule. But there's costs and benefits to this kind of representation. So the kind of obvious benefit of the distributed representation is that it makes it really easy to see commonalities across your experiences. So if you're using this kind of representation to think about the structure of all the different birds that you know about, it allows you to kind of integrate and represent all the kind of common features of those birds, and then generalize very efficiently and easily to new things that you see. So if you see this new bird you haven't seen before, but you immediately can see all of those features it shares with the other birds, you'll be able to generalize and say, oh, maybe that bird can fly even if I haven't seen it fly yet. On the localist side, in the extreme case, it makes it impossible to see those commonalities, but sometimes that's what you want, right? Sometimes when you're trying to remember how things are different from one another, it's useful to use these separated representations to avoid interference. So if you're trying to remember that this bird swims but doesn't fly, even though this very similar looking bird can fly, it's useful to use those separated representations to avoid interference. And this interference difference is associated with a behavioral difference between these two kinds of representations, which is that distributed representations are very sensitive to the order of presentation of information, whereas that's not true over here. So just to give you a quick intuition for this, if you haven't thought about this before, so in a localist representation, you could present information in alternating order, interleaved order, or you could present it in blocked order, so like present the first set entirely before you move on to the second set. And because there's no overlap in that internal representation, it doesn't matter. It's happy for information to be presented however. In the distributed representation, you could do that interleaved learning, and that allows you to build up this nice structured internal representation that reflects the full kind of structure of the input. But what happens when you do blocked presentation is that to the extent that there's overlap in that internal representation, the second population of neurons will tend to take over the real estate of the first set, and you get this interference of those overlapping neurons. So distributed representations are very good at storing structure and data efficiently, but that representation is very susceptible to interference. And this is the classic catastrophic interference problem that many of you have probably heard of. So there are these costs and benefits. What is the brain using? Well, probably it's doing both of these things. And that was really the proposal of the complementary learning systems theory, which said that it's the hippocampus that's specializing in the rapid learning of the specifics of individual experiences using these pattern-separated or localist style representations. And then the hippocampus replays this information offline, maybe especially during sleep, in interleaved order. And that was a critical part of the theory that isn't always appreciated. And that allows the neocortex to slowly build up that distributed overlapping representation that allows you to kind of represent all the structure of the world. But, so you avoid interference here by not writing the information directly to that store, but instead by learning it first in the hippocampus. But there's all kinds of structure and statistics in the environment that we can learn before there's time for all of that replay and offline consolidation and sleep and all of that. So how do we fit that into the story? So we've proposed that the hippocampus is also responsible for this kind of learning, so this rapid statistical learning, and that it's using actually distributed representations to do that. And so the key proposal is that the brain learns distributed representations quickly, and then slowly as you come to ultimately build up that knowledge of the world that's represented in neocortex. So the experiments that got us thinking about the hippocampus as being important for this rapid learning of structure were these temporal statistical learning experiments where participants saw streams of objects presented one at a time, and there was some hidden temporal structure. So in this case, there were pairs of objects that always occurred together in the sequence, and we found that over the course of this learning, objects that co-occurred in the sequence came to be represented more similarly in the hippocampus. And that was also true for this more complex temporal community structure. The hippocampus also represented that kind of structure. And we found that patients with hippocampal damage were unable to behaviorally express this kind of statistical learning. So this made us think that the hippocampus is really crucial for this kind of learning. But then how do we reconcile this kind of rapid statistical learning, building what look like these kind of overlapping distributed representations with the famous history of the hippocampus as being so crucial for keeping experiences separate to avoid interference? So how could one area be doing both of these things? And to try to understand this, we've been working with this model of the hippocampus that we call Seahorse that has pathways and properties that reflect what we know about the actual hippocampus. So there's this trisynaptic pathway, and this is the pathway that's really famous for those sparse pattern-separated representations that we think are so important for avoiding interference. There's this really sparse, specialized connectivity in dentate that leads to this kind of pattern separation function. But then there's this direct pathway, the monosynaptic pathway to region CA1, and the properties in this pathway look different. So the pattern separation is less extreme, and it also has a little bit of a slower learning rate than the trisynaptic pathway. And we've argued that these two different pathways are implementing these two different functions, episodic memory and statistical learning, and that it's these distributed representations that we think might be being built in region CA1 that are really the engine of that rapid statistical learning. So if the model's right, then this monosynaptic pathway learning strategy should support many forms of new structure learning. Really, any time that you have to integrate information across episodes over time, this kind of learning strategy should be helpful. And so we've been thinking about, what are the kinds of learning that this strategy kind of opens up for us, for understanding hippocampal contributions to new learning. So one domain where this is really crucial is when you're building up new categories or new concepts. So this is another situation where you're integrating information across instances in order to find that structure, and we know that the hippocampus has an important role to play in this learning. So we're interested in whether our model can help us understand what's happening in this kind of learning. So I'm going to show you a simulation done by Jelena Sucevic. She's been applying the model to different category learning domains. I'm just going to show you one example here. This is a paradigm we've been using where participants learn about these three categories of novel satellite objects. So satellites in the same category share most of their parts, but they also have unique individuating parts. And we had found in one of our fMRI studies using these stimuli that CA1 represents the category structure. So items from the same category represented more similarly than items from different categories, whereas dentate CA3 doesn't represent that structure. So we train the model on these stimuli, and then we ask it to generalize to novel category exemplars. And the green line here is the kind of normal, intact performance of the model. It can do this generalization better and better with more training. If you restrict the model only to have access to its monosynaptic pathway, it actually does that even better. And if you restrict it to the trisynaptic pathway, it really struggles with generalization. But it's not that the trisynaptic pathway isn't listening to this structure or learning anything here. It is learning something. It's learning about the unique features of these individual objects. So the ability to remember those exemplar unique features is really dependent on the trisynaptic pathway, whereas the monosynaptic pathway struggles with that. So beyond the trisynaptic pathway being important for episodic memory, this suggests that it's important for basically any arbitrary structure learning, whereas the monosynaptic pathway is really crucial in the situations where you need to extract systematic structure across experiences. So this suggests that ability to understand the category structure and generalize really depends on the monosynaptic pathway. And if you look at the internal representations learned by this model, you can see that category structure in CA1 that kind of mirrors what we had seen in the imaging data. So do these kinds of representations that we're seeing in the model manifest in people's behavior? And this is a line of experiments led by Marley Tandock, who's a grad student in the lab, where she's taken these same stimuli, but assigned colors to each of the individual features. So you can see the color arrangement here that's drawn from this two-dimensional slice of color space, where we have this kind of like purple, blue category and then this orange-brown category. And we're gonna see if we can use distortions in color memory as a way of getting at how these representations are changing over time. So Marley teaches people about the parts of the satellites using this feature inference task. They sometimes see the colors of the parts, and we're very careful to kind of make sure they see different part types with equal frequency, the color of those parts. And then she does these assessments of color memory, where she shows people these different options of which of these different colors corresponds to that part. And there's the correct option, but then there are foils that are closer to the category center and foils that are farther away from the category center. And what she finds is that over the course of the learning, the probability of choosing the correct color is the same for the unique and the shared features of these objects. But when they get it wrong differently. So people tend to remember the shared features as being closer to the category center, and that happens less so for the unique features. And this also plays out in generalization, where when people are guessing the color of a new satellite, they'll tend to guess closer to the category center. And the same dynamic. So if you show this exact paradigm to the model, the same thing kind of plays out in the model's hidden representation to the extent that it's driven by that monosynaptic pathway. So we see that same representational structure in people's behavior as we see in the model. Okay, so we're accumulating data that's consistent with this idea that there are these rapidly formed distributed representations in the hippocampus. I think it's, it makes sense. Like, are we done? Have we solved it? Or might there be an alternative here? How else might the hippocampus be contributing to this kind of learning? And actually, there are alternatives that we should be considering. And to talk about this, I'm going to bring up this one more paradigm, the associative inference paradigm, where people learn that two objects A and B go together, B and C go together, and then they're tested on that transitive link from A to C. And we know that this task depends on the hippocampus from rodent lesion studies and from lots of human fMRI studies. So there's different strategies for solving associative inference. One kind of strategy was implemented in this remerge model from Qumran and McClelland, where they said, well, we think what's happening is that the hippocampus is really relying on those pattern-separated conjunctive representations of the trisynaptic pathway, and then computing that transitive link at test. So the idea here is you keep your AB and your BC memories separate during encoding, but then at retrieval, you can still solve the problem by, you see item A, you can remember your AB experience, which gets you to your B item memory, which gets you to BC, and then finally out to C. So as long as you have these kind of smart recurrent connections set up, you can solve this problem at retrieval without having encoded the associations during encoding. That's very different from this other kind of strategy I've been talking about, where you're doing interleave learning to build up these overlapping distributed representations. So the idea here is that you have these overlapping A and C items. As you go back and forth between studying A, B, and BC, there's pressure to kind of merge those representations together because they have that B item in common. And then by the end, you have this direct overlap in these representations, which makes that inference totally trivial because these things are just overlapping. So it's easy to say that A and C are related. So this kind of strategy makes the inference really fast and automatic, but dependent on that interleaved presentation order for the reasons that I talked about at the beginning. As opposed to the localist strategy, which is more slow and effortful, right? It requires you to work through these different associations at retrieval, but it's not dependent on presentation order. It's robust to the order presentation. So we want to use these kind of behavioral differences between these strategies as a way of trying to tease apart what people might be doing here. So this is a line of experiments led by Zi Zhou, who's another grad student in the lab, and he did an associative inference task where participants learned different triads of these items, where sometimes you'll see the A, B, and BC in alternating order, so A, B, BC, A, B, BC, and then for other triads, you see all of the ABs before you start to see any of the BCs. But these are all mixed up with lots of different triads. And then at the end of the training, we do a few different tests. I'm just going to talk about one of them here. The speeded recognition test. This is the one we're most interested in. So the idea is we ask people, did you study these two things together? So in this case, this is an A and a C. And the answer is no. They didn't directly study those things together. But that's a little bit confusing, right? Because they are indirectly related. And our thought was that if people are building up these overlapping representations, which we think might happen under the conditions of interleaved learning, then they should find that judgment confusing, right? So they should be slower to say that the interleaved ACs were not studied together, or they might even false alarm and say they studied those things together when they hadn't. And these are the results across 3 different experiments that had like small tweaks in the design, so they're really just kind of replications. So higher up means a more of a bias for interleaved relative to blocked, which would be down here. And we find across all these experiments that people are indeed slower to judge that those interleaved ACs were not studied together, which is what you would expect if interleaved learning is kind of bringing these things together into an integrated representation. We also found in 2 of the 3 studies this false alarm effect. So people actually get confused and say that they studied those things together when they hadn't studied them together, more so in the interleaved case. So that's kind of an example of a situation where these putative distributed representations are actually not really what you want, right? Because they're confusing you about that recognition task. But of course, we think these representations should be useful. And one of the things we think they should be very useful for is generalization. So we did another experiment where people, again, studied those items in the same way, interleaved or blocked order. And then we taught them that some of the objects, like the A object, had some new property that they had to learn. And we tested how likely are they to generalize that property to object C. And we find that they're much more likely to do that in the interleaved case, suggesting that it's kind of benefiting learning in that way. We also have an experiment I'm not going to talk about on the benefit of interleaving in statistical learning. So if you're interested in that, you can check out the paper. But overall, we think that this line of studies suggests that interleaved learning indeed quickly leads to these signatures of distributed representations as we'd expect from the model. Okay, so far I've talked about this kind of direct learning from the environment, whether you're building kind of pattern-separated representations or integrated representations, but ultimately we think that we're trying to build up this highly distributed representation of all of our knowledge of the world, and that there's something happening offline that helps make this happen. So even without further experience directly from the environment, we're somehow able to do this transformation where we build up these representations in neocortex. So how does this happen? So I'm just going to briefly tell you a little bit about some of the work the lab is doing to think about this kind of side of the kind of memory acquisition process. And one prediction of this framework, this way of thinking about things, is that you should actually have a better understanding of the structure of your environment after consolidation, in particular after sleep, when we think a lot of this work is happening. You should building up this kind of overlapping representation should lead to a better understanding of how things are related, even without further direct exposure to that information. So we tested this using those same satellite objects that you've seen, where we found that over a night of sleep, people's memory indeed improves above baseline for the shared features of these objects, suggesting that there's something happening that is allowing you to better understand that category structure. So how does that actually happen? Like, how can the hippocampus and neocortex interact on their own without input from the environment and do some useful learning that actually results in that kind of memory change? And to try to think about this, we've been working with a sleep model. So it has a kind of a simple version of our seahorse model connected up with a big neocortical module. And I'm not going to go into the details of how this model works, except to say that it runs completely autonomously, so it's able to move from memory attractor to memory attractor on its own. It does kind of concurrent hippocampal cortical replay, and it knows kind of knows how to find the right moments to learn from those memory attractors offline. Oh, and I should say this is Daria Singh's work. This is a PhD student in the lab. Okay, so Daria, again, applied the model to these same stimuli and lets it run, so first it does normal awake kind of training, and then it runs completely on its own offline, and we find that the model does uniformly interleave its replay of these objects. So this is kind of the transition from each object to each other object, and you can take my word for it that that's uniform. And we find that this results in the kind of integrated overlapping representations that we'd expect of those categories in the neocortical module. And that this also results in that behavioral improvement in the model in the shared features, like we had seen in our human data. So it suggests that it's capturing something. It's one way of capturing what's happening in that data. The model also has sleep stages, so it has a period of time that corresponds to non-REM sleep, where the hippocampus and cortex are very tightly coupled in their interactions. We know that there are these coupled oscillations between hippocampus and cortex that we think are facilitating communication between these regions during slow wave sleep or during non-REM, so we have that. And then we have a REM stage where things are less tightly coupled, and we let the neocortex kind of run more on its own and explore its existing attractor landscape. So our thought here was that these different sleep stages, and in particular alternating between them over the course of a night, might be really important for graceful continual learning. So the idea here is that you learn some environment one, so you learn some information that you can imagine this as like a very remote kind of fully consolidated bit of information, environment one. Then you start to learn some new environment two that's going to be related to environment one. And the question is, how do you kind of avoid the kind of interference that we've been talking about when you're learning environment two, retroactively onto environment one? And we do 3 simulations, so the model is going to see environment 2 for a little bit, then it's going to sleep either with only non-REM sleep, or it'll alternate non-REM and REM, or only get REM sleep. And what we find is that, so over the course of a night of sleep in this model with these alternations, you can learn environment 2 from either non-REM by itself or by alternating non-REM with REM. But if we're asking what happens to that remote environment 1 knowledge, the only way to retain environment 1 while you're learning this new environment 2 is to alternate over the course of the night. So basically what's happening is that non-REM sleep is focusing on the more recent information. It's more hippocampally driven replay or dominated replay. And then during REM sleep, you have the opportunity to go back into your existing cortical knowledge, pull back up that old knowledge and kind of prevent it from being overwritten. And then you're able to kind of integrate that new and old gracefully. I thought this is floating around Twitter today, so I just thought I would advertise that we're also interested in sequential replay. So what I just showed you was all kind of static replay of individual objects, but we're also very interested in sequential replay and engaging with the kind of like rodent literature on forward and backward replay and all of that. So that's a paper you could check out if you're interested in that. Okay. So the hippocampus, we think, might contain these distributed representations that complement the kind of classic pattern-separated localist representations, and our theory is that that might be occurring especially in region CA1 on the monosynaptic pathway. But that this is part of a broader process, maybe even we could think of it as a continuum, where during sleep, the hippocampus helps the neocortex to further kind of enhance that shared structure, and then crucially also to integrate the new with existing knowledge into those kind of highly distributed representations that reflect your full knowledge of the world. Okay, here, we're looking for a postdoc and an RA. We just got a R01, so please come join us, and thank you so much for your attention.